Our final presenter this morning is Chris Smillie from Massachusetts Institute of Technology. His field is biology and computer science and bioengineering under advisor Eric Alm, and his practicum is at Lawrence Berkeley National Laboratory.
Hello? Oh, okay. Oh, sorry. Yeah, so I'm Chris Smillie. As he said, I'm in Eric Alm's lab, and we study the collection of bacteria that live on the human body, which is called the human microbiome. So first I thought I'd give you a quick, there's a few intro slides, and then I'll talk about one small project I did in the lab. I don't know if this is comforting, but you are not alone. Ever since birth, you've been colonized by trillions of bacteria. And with the effect that, like, now for every human cell in your body, there's 10 bacterial cells. And for every gene in your genome, there's 100 bacterial genes. And even though they're numerically dominant, because bacteria are super small compared to human cells, they're like roughly 1 to 2 kilograms by weight, so like the size of a small watermelon or something. And the good news is these bacteria are, like, they're good for you. They influence your health. They help you metabolize food. They help you metabolize drugs. They protect you against pathogens, and they help coordinate the development of your immune system when you're young. When these bacteria sort of get messed up, and they can change into sort of a more unhealthy state, and they can cause disease. So the human microbiome has been associated with obesity and malnutrition, autoimmune disease, acne, colorectal cancer, and many other diseases. So how do we study the microbiome? Usually we take, well, I can't point here, but usually we take some sample of bacteria, for example, your plaque. We break open the cells, extract the DNA, and then we chop up the DNA into small fragments and sequence it. And this tells us what bacteria are present and what genes they contain. And so it's, it's sort of quantitative, but not super quantitative. But this is the data that we work with. And so now I'm going to tell you about a project that I've been working on. It involves a Clostridium difficile infection. And Clostridium difficile is it's a pathogen, and it's the leading cause of antibiotic-associated diarrhea. Roughly 500,000 people get sick from this each year, and I guess around 15,000 people die each year. So it's a major problem, and the way it works is that usually somebody will go into the hospital for some completely unrelated condition. They'll take antibiotics there, and this wipes out a lot of their healthy bacteria in their gut. And if they have C. diff in their gut as well, so after killing all the healthy bacteria, C. diff activates, and it starts producing a number of toxins which trigger an inflammatory response, and this leads to sort of a persistent state of dysbiosis where the patient is sick and has lots of diarrhea, and their life is like basically miserable. So 70% of the time, and the first line of treatment is vancomycin and metronidazole. These are 2 antibiotics. And 70% of the time, that's enough to cure the C. diff. 30% of the time, these antibiotics, you're just wiping out the healthy bacteria again. And then, you end up in this infection cycle where you just, you treat it with antibiotics, but then that helps it, persist in your gut, and so on and so forth. Recently, like within the past 5 to 10 years, there's a new treatment that's available, and it's very effective. What you do is you take stool from a healthy donor, and you put it into the diseased person's gut, and this is called a fecal transplant. And so fecal transplant is very effective. It successfully cures C. diff around 90% of the time. And so there's a lot of interest in this treatment. So aside from C. diff, people have been using fecal transplants to treat a number of other diseases. So big ones are inflammatory bowel disease. People are interested in treating obesity, malnutrition, hepatic encephalopathy, and a number of autoimmune disorders. Right now, like you just have a stool donor and you take stool from them and you put it into the patient. That's like a little crude, you know. And eventually, I think there will be pills that people take to treat these different diseases. There are a few like interesting anecdotes. I guess I have time. So like sometimes people have C. diff, but they have other diseases. So one interesting case is when somebody had C. diff and alopecia, which is where an autoimmune disorder where you lose your hair. They had a fecal transplant. It treated the C. diff, and it also cured their alopecia. So people don't really understand what's going on, but it's pretty cool because, like, you know, this is a completely new drug, and we just don't understand how it works. So for this study, so I was lucky enough to be involved in this study. We collaborated with Dirk Jeevers and Bruce Buren at the Broad Institute. And on the clinical side, Libby Holman, Elon Youngster, and Ginny Sock. And basically, we enrolled 4 healthy donors and treated roughly like 20 C. diff patients with fecal transplant. And in the end, roughly 90% of the patients were cured. So blue is healthy and red is diseased. And what we did is we used DNA sequencing to sort of measure their microbiomes before the transplant, after the transplant, and we also measured the donors. And we're really interested just to know what's going on. So this is what it looks like when you get a fecal transplant. This is the top row is the patient before the FMT. So FMT is fecal transplant, just so you know. So top row is the patient before the FMT. The second row is what the donor looks like, and the third is the patient after the FMT. So white so each column is a different type of bacteria. White means that bacteria is absent. Blue means that bacteria is present, and you know the shade of blue, darker blue is more present or something like that. The point of the slide is just to show you that if you look at the patient and the donor, there's really like no way to predict what the patient's going to look like after the fecal transplant. It's not like some obvious combination of the two profiles. But we're interested to know which bacteria are going to survive, which bacteria will be successfully transferred from the patient to the donor, and what are the factors that sort of determine this engraftment. So if you do something pretty naive and simple, and this is a phylogeny, it just depicts the evolutionary relationship among different bacteria. If you do something pretty simple, you can compare the bacteria before and after the transplant. Blue is enriched after the transplant. Whatever this color is enriched before the transplant. And you can see like there are some clear phylogenetic biases and things like that. But this is just like a really coarse view of the data. What we're interested in doing is trying to build a statistical model of the fecal transplant itself. So we took lots of different data. We know the abundances of the bacteria in the donor and the patient. We have a lot of clinical metadata. We have a lot of phylogenetic data. And so we used all of this and we threw it into a model, just like a random forest, which is a machine learning approach. And so this is, on the x-axis is the true abundance after the FMT, and then on the y is what we predict. And so we can get like this really good predictive model that basically tells you, given a donor and a recipient, what's going to happen to that recipient. So we're interested in understanding what factors are sort of driving these predictions. If we look into the model, basically what's important is just the bacterial abundances and some phylogenetic effects. But what's interesting is those are the main factors that are important. This is a different view of the data, sort of. So if we look at the input bacterial abundance, then the probability of engraftment goes up as the input abundance goes up, which is cool. And same here, so input abundance goes up, and the abundance after the FMT also goes up. So this means that higher abundance bacteria engraft more frequently and in greater amounts, which is like maybe not too surprising, but that's useful for people who want to, oh my god, who want to develop synthetic sort of like FMT pills. We also looked at the phylogenetic effects. So after controlling for abundance, different phylogenetic groups of bacteria are enriched or not enriched in FMT. What's interesting is that within a group of bacteria, you find some that have a negative effect on FMT efficiency, and some bacteria which have a positive effect. So we're currently looking into that. So our model, this is the same thing as before. Our model explains a lot of the post-FMT diversity. These are our predictions in the bottom row. So you can see that there's a lot of agreement. But what I actually want to talk to you about is some more parallel computing stuff that I did. And so everything I've told you so far is at the species level. And maybe you won't really appreciate the significance of this. We're interested in looking at a higher resolution view of the data at the strain level. So the reason this is difficult is because strains look very similar at the DNA level to each other, but they differ in these positions which we call SNPs. So blue are where they're the same, and red is where they're different. The problem is we don't know which SNPs go together, which SNPs travel together, because we've fragmented the DNA and we've lost that information. So you can imagine that if you put 3 different strains into the sample at different frequencies, you can look at patterns of the SNPs. So you can look at the frequency patterns of the SNPs and try to sort of like figure it out and reverse engineer what the strain genotypes were and what their frequencies were. I'll just sort of skip this, but this is just like a more mathematical view of the problem. So we look in our sequence data. The way we sort of figure this out is we can calculate the likelihood of our data given guesses of the strain genotypes and the strain frequencies. I don't really have time to go into this. This is the likelihood function. It's hard to directly optimize this, but we can fix the strain frequencies and optimize the genotypes, and then fix the genotypes and estimate the frequencies and just iterate back and forth. And so this is the approach we took. I'll skip that. And then this is validation of our approach. So we tested it in many different scenarios with many different combinations of parameters. And this is just an example, but it shows you that our estimates of the strain abundances, these are the true ones, these are the estimated ones, and they agree pretty well. So I don't have like tons of time, so I'm just sort of skipping through this stuff. In the end, so after using this method, we find some interesting things about the fecal transplant. We find that in general, the bacteria that are in the donor are really important. And so the patient ends up actually looking like the donor. And this is interesting because different people have different sets of bacteria which may influence different phenotypes, like your BMI or something like that. So potentially, if you have like a high BMI donor and you transplant that into a patient, then that patient might inherit this high BMI. But this is all like super speculative, and it's just one thing. So in general, the strain composition is often conserved between donors and their recipients. And we also find examples of new strains coming in. And in cases where you have competition between the recipient and the donor bacteria, we have a mix of outcomes. So sometimes the recipient ends up looking like itself before the transplant, and sometimes it looks like the donor. So this is sort of like a work in progress, I would say. And in general, we can sort of like look across all of these events and figure out like, so it looks like roughly 40%, in 40% of the cases, the donor strains actually engraft in the recipient. And like 20% of the cases, it looks like the recipient before the FMT, and then the rest of the time you get environmental strains coming in. But so that's it. I'd like to thank a few people. Everybody in the Alm Lab, our collaborators at the Broad, our clinical collaborators, and especially the DOE CSGF. So thank you, and I can take some questions.